right? So this is very important. So listen for a moment. It is supposed to identify every single bioactive genetic sequence that is in that piece of bacterial DNA. And so this is a perfect example. Remember I said it's like our DNA, which is linear, but it's joined at the end, so you get that circular DNA. That's why this is shown as a circle. And you can see all these little lines with all these little designations, right? What those are all telling you is those are all pointing to bioactive segments of this bacterial DNA, right? Pieces of genetic material that if it gets into a biological organism, it can do something, is what that means. That's what it means when we say bioactive. Now, what is completely unacceptable would be to fail to first of all, to fail to disclose any bioactive genetic sequence, especially if that genetic sequence could be active in what we call mammalian cells. Why? Because we're mammals, and so all of our cells are mammalian cells. So for example, some of these genes can only be expressed in the bacteria, right? Many of them are designed, it's bacterial DNA. So it's designed to be used by bacteria. So a lot of these can't be expressed in a human cell. But you can imagine, therefore, that it would be particularly egregious if you do not disclose any sequence that could be active in a human cell. And what I want to point out to you is just one thing on this plasmid DNA. So this was confirmed again by David Speaker. And there's one in here, it'll be hard to see, but there's one line and it points to, yeah, so right here, what I'm gonna, if your readers look on the right-hand side, look at that cluster of labels on the right-hand side and move up just a little bit to the top, you'll see the SV40 promoter. That's perfect. You'll see it with a circle around it, right? It's highlighted, SV40 promoter, and you see that line pointing to where it is. So in other words, so what they did is when they sequenced these bacterial plasmids, they found the sequence in there. Now, what is critical for your readers to know is Pfizer submitted a plasmid map. As they were required, they submitted this to Health Canada. And you know what was amazing? When Kevin McKernan and then when David Speaker got this data as well, they went back and confirmed and double checked. They put this side by side with the plasmid map that Pfizer submitted to Health Canada. And that SV40 enhancer was not there. It was not disclosed. And what's interesting also is there's a second very large fragment. We've been focusing on the publicly on the SV40 promoter, but there's a second very large genetic sequence that's in there as well that has not been disclosed. And based on evaluation by molecular biologists, it appears that that undisclosed sequence also has the potential to be expressed in our cells. And we have looked. We have no idea what that genetic sequence is. It doesn't match with any known gene in the human genome or any other gene from any other species that we've seen so far. But it's in there, and if it were to be expressed in our cells, it definitely wouldn't be recognized as self because it's not in the human body. So that would be recognized by the immune system and make cells a potential target. And we don't know what other things it could potentially do. And it's a very large sequence. And now, just before we move on with this, Laura Lynn, what you have to understand, this is so important. These sequences are relatively large. Pfizer disclosed to Health Canada bioactive sequences that were as little as what we call 7 base pairs. 7 base pairs. All right. These fragments that I've just been telling you about that were not disclosed are far larger. So they went into minute detail to reveal genetic sequences. They left these ones out. The second thing I want you to know, Laura Lynn, is that map can be automatically generated. So for example, my lab uses what a lot of labs use for this work. It's a piece of software, we call it SnapGene. If I imported that file that Pfizer had provided into this software, it will automatically do what we call annotate it. It will annotate it. It puts these labels on. It will automatically, so Kevin McKernan did this with that software, it automatically annotated, as it did for, as you can see here, for Dr. David Speaker, it automatically put on that SV40 promoter label. It didn't label that other sequence because nobody knows what it is. The software doesn't know what it is because it looks like it's something that hasn't been discovered before. But the point is, at least the SV40 promoter automatically gets loaded there. So again, we don't see how this could have got to Health Canada without somebody actively removing that label, because we're not aware of software that Pfizer would possibly have been using that would not have automatically labeled this. And the reason why this is important, so they know, this is the other thing that's very important, that SV40 promoter is not needed.